Okay, hello everyone, welcome back. Today we're going to talk about part 3C of sugars and nucleic acids, and this is going to focus on short interfering RNA technology. All right, so we're partway through, as you can see through the outline here, and short interfering RNA technology, what is it and how does it work, and how can we use this to develop therapeutic oligonucleotides? So short interfering RNA technology involves long double-stranded RNA, and this could be something that is transcribed by the organism itself, or perhaps a virus, which is comprised of double-stranded RNA, infects the host. And these double-stranded RNAs are quite long, and they are cut up by an enzyme called dicer. So this is an enzyme that cuts the long double-stranded RNA into something called short siRNAs. siRNAs means short interfering RNAs. And these are typically 21 to 23 base pairs. And what can happen is that these siRNAs are a substrate for something called the RNA-induced silencing complex. Also abbreviated as RISC. And one of the proteins within RISC is called argonaute, as shown here in this cartoon. So argonaute will recognize the double-stranded short interfering RNA, and what it will do is it will degrade one of the strands. So this is sort of a very simplified version, but what's going to happen is that one of the strands is going to leave, and then it's going to be able to have base pairing complementarity to a messenger RNA. So once it does that, you can see here in this light green is that you have this degraded RNA, and argonaute 2 is catalytic. It's able to degrade RNA. And if this is your messenger RNA, then that means that you can no longer make that protein. So what's going to happen is that the degraded messenger RNA is going to be released. No protein can be made. And now you have, again, an active argonaute complex that can now seek out another messenger RNA and target that with base pairing complementarity. So you can think of it sort of like a catalyst a little bit. So it seeks the messenger RNA over again. It's like a catalyst. And it's very efficient at cutting that specific messenger RNA so that no protein can get made. So you can think of this as a mechanism in place that our cells use in order to control the level of certain genes. And this offers one mechanism in which we can do this. In 1998, researchers Fire and Mello discovered and reported that an organism called C. elegans, which is a worm, can utilize double-stranded RNAs to control gene expression, as I'm showing you here. And as I mentioned, this is an endogenous process, but what is particularly exciting is that you don't have to use long microRNAs or viral RNAs. You can actually use a synthetic siRNA that is made in the laboratory. And is it possible to interject it at this point here in order to generate a synthetic siRNA that could target potentially any of the 30,000 genes? So recall that we have 30,000 genes, and only about 15 to 20% are thought to be druggable using small molecules. So using a synthetic siRNA, we could potentially target any of those genes that could be implicated in things like diseases. So let's look at some of the modifications that have been used for siRNA. And as you can imagine, RNA is very nuclease labile. Nucleases are able to cut that phosphodiester bond, and therefore there needs to be a mechanism in place in order to give it inherent stability. So a number of different modifications have been used over the years. I'm just going to highlight some very common ones that are prominent in the field. Here you can see you have regular RNA structure, so you're familiar with this 2' hydroxyl group. As we've looked at in course pack, this adopts a C3' endo conformation as a dominant conformer. However, as you know, you have the phosphate, and then this could potentially act as a nucleophile in the presence of base or nuclease-mediated catalysis. So in order to strengthen the lifetime of the RNA, modifications like O-methyl have been used, and these have been quite robust. This one here is called locked nucleic acid. So you can see you have a bridging carbon between the C4' and the oxygen here. So we call this LNA, or locked nucleic acid. And what this does is this actually keeps the RNA in only the C3' endo conformation. So it can't flip out to a C2' because it's actually in a constrained ring system. Some other interesting modifications, this is a 2' O-methoxyethyl group that we studied earlier. This had some utility with things like antisense, and this here is a fluorinated derivative. So these are just some highlights. In addition, researchers have also modified the heterocyclic base. You can see here, this one is called 2,4-difluorotoluol group, and it cannot hydrogen bond to its partner. Yet, if you place this within siRNAs, you give it different properties, and you can fine-tune the structure-activity relationship with these types of modifications. So here we have sugar modifications, and this is a base modification. And again, all of these modifications are done at improving things like stability, cellular uptake, where it goes in the cell, so the biodistribution. We want to inactivate, or we want to make these molecules not immune system active, so we don't want our immune system to target these molecules if they're a potential class of therapeutics, and then furthermore off-target effects, so targeting similar genes. Sugar, base, and then finally the backbone is another site for modification. So here's that phosphodiester bond that I mentioned to you. It's quite labile. So researchers have placed phosphorothioase, which you've learned can give the molecule inherent stability. Here's an example of a boranophosphate, which still retains a negative charge, but can give it some different character as well. In general, there has been fairly little explored in modifying the backbone. However, in recent years, there have been efforts to synthesize derivatives containing neutral backbone. So things like amides. I've done some work myself with triazoles. So this is an active area of research in the field. There are several companies on the horizon looking at siRNA as a potential platform for curing disease. And this includes companies in mainly the Boston area, some also in California. There's a company in Vancouver called Arbutus Biopharma Corporation, but largely the company here, Nyland, has been a real driver in siRNA development, and they actually have 2 siRNAs on the market. So this is a figure from Varova and Watts in 2017, just highlighting the discovery of RNA interference, which I described, and where the field is now, and kind of gives you an idea of optimism and where things failed, and where we are now. So as I mentioned, in 1998, Fire and Mello discovered this RNA interference, and there was a lot of optimism in the field. So you can see here the market size, optimism, visibility. I'll also include things like market investment and funding, literally skyrocketed. In 2003, 2004, there was some promising clinical trials in the field. And in 2006, only 8 years after the discovery, Fire and Mello received the Nobel Prize for the discovery of RNA interference. Big Pharma entered, okay, and however, around 2009, during the economic downturn in the United States, and we felt that here too, several advanced clinical trials failed. A lot of big pharmaceutical companies left, market investment left as well, and then there was really very little optimism in the field. It wasn't until around 2012 or 2013 when new chemistry showed that they could be used to improve the properties of these siRNAs. So one of the main issues was that it was really hard for these molecules to get across the cell membrane. So delivery was a major issue. And what researchers did was they used something called a lipid nanoparticle, which I'm showing you here, which is essentially a synthetic micelle that encapsulates the siRNA and helps deliver it across the cellular membrane. So these lipid nanoparticles unfortunately showed immune effects until 2011, 2012. So they weren't properly being utilized in the body. So some modifications to these lipid nanoparticles and delivery system. We'll talk about this molecule here called GalNAc siRNA in the next class. Really expanded the delivery and allow for renewed optimism in the field. But I must tell you though that most people and most companies abandoned siRNAs as a potential therapeutic, and it was just a few companies that were really driving and working extremely hard to make this happen. And in 2018, a compound called patisiran by Nyland was FDA approved, and this is actually the first US FDA approved siRNA on the market. And then a year later in 2019, another siRNA called givosiran was approved by Nyland, again, targeting specific disease. And different companies like Quark and Ionis have other types of candidates in clinical trials. So right now it's 2020 and things are certainly very exciting in the field for siRNAs. Let's just take a quick little snapshot at Nyland. This is a screenshot from their webpage. So this is 2020, and it keeps, I encourage you to check it out if you're really interested in this stuff. Onpattro, as I mentioned, is the first siRNA, Givlaari or givosiran. These target some rare diseases. One of them is called hereditary ATTR amyloidosis, and then this Givlaari targets a liver disease called acute hepatic porphyria. There are definitely a lot of other candidates in clinical trials, as you can see, and these target a lot of diseases like metabolic disorders. This is hypercholesterolemia. Fitusiran, this targets rare bleeding disorders. There's other things like alpha-1 liver disease that is in sort of early stage. And then there's RNAs that can also target not just our own genes, but things like viruses. So like a hepatitis B virus infection could be very useful since those viruses are comprised of RNA. Hypertension, which is a leading cause of death in North America, is another very important strategy as well. I just want to highlight to you this one here. This ALN-CoV targets the COVID-19 coronavirus. And this is, of course, an early stage because it's 2020. This pandemic really just started. But they have some candidates where potentially you could be given an siRNA, and you could have a sort of a respiratory nasal spray similar to something like an asthma puffer, and this would go into your lungs, and this would really help eradicate the virus. So there's a lot of excitement in the field and a lot of versatility too in using these types of molecules. I'll just highlight a little bit about one of the molecules. The first one, called Onpattro, how it targets this rare disease. So I just want to talk to you a little bit about the mechanism of how this works, and this will be the last slide for this unit. So again, this is called TTR amyloidosis, and TTR stands for transthretin, and this comes from these words here, where it transports thyroxine and retinol. So it's a protein that transports thyroxine and retinol, which are important molecules for the body. So it turns out that a single mutation in TTR protein can cause this protein to actually form fibrils and aggregate and form these like stacked clumps. And if this happens within your organs, this can be very painful, and it can lead to toxicity and essentially organ failure. So one mutation in TTR causes TTR proteins to aggregate and form insoluble fibers. So you can see that these misfolded chains are now stacking up onto each other, and then these amyloid fibrites form these ordered beta pleated sheet structures in these tissues, and because of this deposition within the organs, this can lead to issues in the heart, renal failure, nail dystrophy, very soft tissue. So it's a rare disease, but it can be very painful and very toxic, especially when it's targeting the heart. So the siRNA patisiran actually targets the mutated form so that you don't get these fibrils forming, and thus you don't have the phenotype of this disease. So very exciting. And as I mentioned, this was only discovered or put on the market 2 years ago. Okay, so that's what I wanted to mention today for siRNAs. We'll definitely talk more about siRNAs if you take the advanced topics in chemical biology, Chem 4120, next term. Okay, everyone, enjoy your day and see you soon.